They didn't show up?
They do.
Oh, I think it was a group. Oh,
Oh, you mean a meeting? I'm only meeting one person. And the pedigree, and I'm meeting her in front of the vending machines. Yeah, I haven't even started thinking about the group project. Oh, I think there's something due at the end of the month, like who's doing what, but But then I kept, when I was supposed to come back after.
Same here. I haven't looked. Didn't fold up. Yeah, that's why I thought, and I didn't realize what I did. Yeah, we have to fold up back. Right. Also, the other thing I wanted to say, I don't think it was like a tax thing for Ireland. So if you could ask her when you drop her to Russia, I will when I could pick her up. If as long as she has it before Friday, it would be good when I will pick her up. And what's the last thing I wanted to ask? There's something. You know how like every. And then the final thing was did you check on Riley? She's cold. Okay. Right. Alright. Alright. Yeah, I'm gonna tell her. I'm gonna tell her. I'm just eating lunch, so I'm gonna inhale my food because I want to be done by the time class starts, so that I can take a few minutes and just actually digest it without like feeling like it's gonna come back up, which probably taking it slower would help, but yeah, okay, so I guess let me know when you look before options. Alright. Love you.
Not like last year, 'cause I did it. That's like 2010.
Oh, okay. You were.
Yeah. Where there's just a comeback earlier. And I did. Like I deferred for a year, and then like after I was like, oh, one more year, and then just come back. So
So
then
you try to be a
we did it.
because I'm like, I said you do, but I was wondering.
No, yeah, I did it. I feel like I'm not sure. I'm not sure if I care. Like, I don't know.
or like they put you into position because they accepted you first time.
Yeah.
That's a good thing.
I'm not
No,
sure.
I don't want to take a break. I want to be done.
Yeah.
Experiencing her. So did you work in that pizza?
I worked a few, not like super high level,
Okay.
so it's not like really complicated cases.
Okay.
I kind of like it that way.
Yeah. Do you think your experience is going to be of value or anything?
I think it is because but maybe not like exactly what you see, but maybe sometimes that too. I think it's just more being okay that you don't know something, and then
Knowing what resources to
or just asking someone.
Right.
I feel like sometimes I think when you like first come to anything, you feel like shy or like shame about it, but then like you have some experience, you see someone
You may see something that you never seen.
yeah. So I think it's more like the things like that.
Yeah, maybe that's what I'm looking for because she's like a strong believer that, you should just go straight through the ETP.
Yeah, because it's the same.
Um,
I think if you have those personality traits, that's fine. I don't, I guess, not really, not until like
Right, and I'm not, and I'm going straight through it right now, but I feel like even after I'm done, I probably will work as a nurse for a year, even though she keeps saying we shouldn't, unless something really happens where I don't do that. But
well, I think if you like go through this program, then you can just
skip that.
you don't have to be a nurse. It's just
You think I could skip it, still
yeah,
have that
because it's like
confidence.
like in terms of residents, or they don't know anything. They still present. And she'll like ask.
But I think the difference is that for someone like you, if you finish it, are you working now?
Yeah, I'm still working now, but.
So, because from someone like that, I see like if you finish your residence, and if you finish the entry phase to the focus, and you want to go work in the hospital,
Oh, in that.
that's probably easier if you want to work. Like, if you just want to go to primary care, you just want to go to primary care, and you're doing this and not really worrying about it.
Do you want to be in the hospital? You know for sure.
I kind of want to, a little bit upset, so it's really making me consider it. I want to play. No, I thought I was trying to break earlier, but no, I'm saying like, wanting to work in a hospital that I might have to, even though I don't have my NP and for a year or so as a nurse, and I would be able to use my NP if I wanted to take the hospital route, because I don't see how else, I mean, a lot of the hospitals and a lot of people, I guess
Talk to her?
Huh?
Did you talk to her? Because I don't.
She's more kind of black. I have some
But does she think you probably don't need it, right? To go to the hospital?
I mean, she's been clear that she raises primary care people. That's what her essence is. So if you want to work in a hospital, it's not that you can't.
Yeah,
It's that
I feel
you're
like
going
she would
to
figure it out.
You have to figure it out. You're on your own. That's what I got from her answer.
I think if you because if she knows that if she gives you like a lot of clinical sites in the hospitals, you should like network there.
Right, exactly.
I feel like when I was in TP, like even though it's really short, like I could have done it a lot better. Like right off the bat,
Right.
like talk to them, even if you don't know if you want to work there,
Right.
make sure they know your name and
Right.
yeah, stuff like that. And I was like, oh, I'm shy, or like I don't know, or I feel like not confident.
Exactly.
But like, I guess when you're there, they're too busy to really care. But if you stick out, they'll like notice you, but they don't like.
They don't make an effort to like, oh, I want
They
to
won't
work.
like make
It's
you
busy.
interview.
Yeah, so you're right about that. So I mean, for me, like I decide, as I go forward with this phase, I will determine like, I really want to do the hospital now, but the way things are shaping Yeah, it does. If I had like two, it's more like I need more.
Yeah, it messes me up that class is like right where it is. If I just yeah, no, what I just did actually is I met with my partner for the first time ever. I didn't even know she was going to teach ETP like out here. Tiffany saying, oh, you know? Oh, okay, yeah. See, like I was telling her, I was like, if you weren't in like the little one, you can sit up in the front, then I have no idea what you sit here. So yeah, basically, I just touched base with her to see how she wants to do it. Like we're gonna touch base again in July because right now she's studying for class. So she's like, this is not even in my priority right now. I was like, okay, that's cool. So well, but like, is it the week after? Because the way I understood it was that, and maybe I'm wrong because I also didn't ask. But I thought we had to wait for the first pedigree to come back and for the second one was due. Like so that we could see our mistakes. Now, whether like I don't know what the date is, so I think it's totally up. I don't understand why we can't just end it all together. Because then I also understood, or I heard that the actual narrative isn't due until the end of the semester. Which, yeah.
Oh yeah, that's the last day. Yeah, that's the last day. And then yeah, goes to her house. Yeah. I wonder what yeah, she hasn't told me the date. No, she hasn't said that.
Because it seems 1 through 10,
Right.
but you don't know when. Obviously, if I'm 10, I'm going to last. But when?
Yeah. And then
That's
at some point this month,
that's why she goes through it different.
That's true.
Because she said she wants to see this little sort of way she's gonna go over it. But I hope that is like.
oh, it's the 27th of this month. Yeah, so that means sometime this week I have to email. Oh, that means sometime this week I have to email like.
So that was the when because that means a lot of now. You gotta research that and figure out what you want.
Oh Lord. Alright, I guess I'm emailing my proof today. Oh, if you go to the main Columbia website, there's like a search. And then if you just type in the person's name, it'll do like a search in a regular whatever document name, person's name. And I show you Hey, how are you?
Well, I just wanted to share with you that I have sickle cell, and I usually get accommodations for my classes. I don't think I'm gonna need it for this class. But I just wanted you to be aware,
Oh, of
and
course.
if you needed to like see the document or whatever,
Good. Yeah.
I don't think I'm gonna need any of the
Well,
accommodations for the class. But I just
If you
wanted
miss
to-
one or two, I'll make an just don't make it public. I'll make an exception. That's fine.
I just wanted
Are you
to-
okay? No, it's fine.
Keep hanging in there. You know?
Okay. Okay.
All right. So you don't
Don't-
need the
no.
forms? Okay.
I believe you.
Okay. I just wanted to
Okay.
make you aware. Thank you.
Don't try to be a martyr. Like if you need to stay out.
I'm one of those. So I
Okay. Okay.
sometimes, but
Just take care of yourself. Okay.
all right. Thank you.
Okay.
You all
I'm
set?
good.
All righty.
I try to do it by myself. You know that.
Doing good so far.
All right.
email you that they wouldn't be here? Did anybody email you? Two people? Who? Casey and then Daniela? Okay. I just want to make sure I got everybody. Hi. How are you? They're posted online.
I feel like it's the one time I've given a lecture and it hasn't been raining. Like every
I know,
time
right?
I've been running
I
over
know,
and
it's beautiful
raining,
out
so it's
finally.
nice, yeah.
It's a big class this time.
Compared to last summer,
I
yeah.
know. I really appreciate you doing this.
I enjoy it. So.
You do a great job.
Thank you.
I have all these boxes in my office. I'm moving, and so I just brought one little one with me because I don't want to look like I was homeless.
Are you moving to a better spot or just a smaller spot like that?
I am moving back to North Carolina.
Oh my goodness.
But I'm still going to be teaching genetics. We're going to work it so that most of it's online, and then for like these lectures, I'm going to have a TA here to like greet you guys. So I'm moving back home.
Are you just going to do this part time and do that, or are you?
Well, I'm giving up my clinical position here. I'm just going to be doing the genetics for the doctorate and the master students. I know her. How are you, darling? months because you'll just do it and then wouldn't have anything else to do for the rest of the summer. I don't want to make it work. So next week, your first pedigree due on did everybody get the memo for the cystic fibrosis? The guy really wasn't attending. That would be a cool case study though. He was really 30, 40 there when make it legit. Any questions before our speaker gets started?
Just to clarify that, pedigree is on our...
Yes, it's your pedigree. And then your partner one is due a little bit later. This is a very compressed semester. I'm sorry, I usually spread it out a little bit more, but we don't have as much time. Yes, ma'am.
Do you want the pedigree handwritten or do you want online? But it's like,
You can do handwritten. If you do handwritten, don't do like cross throughs and stuff like that. Some people use there's actually a free pedigree software that you can get it. Oh, well, it's not pedigree per se. It's called RDF. RF Flow, that's one I use. It's free. You can get the trial version for free, and then you can draw it if you're into the free stuff, or if you want to use it for your clinical stuff. Formally, it's RF Flow. You just do the trial version, and then you can delete it off your computer and get it free again. Or you can just do it and you can do it freehand. If you have a complicated pedigree ever, you can actually get pedigree software that will let you do little legends and stuff like that too. But for this class, you can do it by hand. If you do it by hand, if you turn it in with all kinds of crosswords and stuff like that, I'll give it back to you to do it over because that's not very good. Okay. Question over here.
Wait, so a personal pedigree too, and then so that when you set this,
The first pedigree is the one that's your family history.
Right, okay.
Yes, that's your family. That's why I do it. And then the second one is your partner's. The questions for the paper is based on your mock, and that's due later. I think at the end. Yes.
You're supposed to download them and scan them in, or do you just bring it to class?
You just bring it to class.
Okay.
Yeah. That would be easier. And then while we're doing the lecture, I can actually I'll grade it for you guys. So when if you bring it at the beginning of the class, I'll grade it during the class. Or if you get it at the end, I'll give it to you the next week. Yes.
okay, so for the team project, will we get like a timeline as to when each set of teams will be presenting?
Yes. I'm going to have that posted by next week. I'm giving you guys some time to get everything together, and then I'll post that next week.
But we just have to let you know about the responsibilities.
Yeah. Yeah. There are two students who need to go on the 17th. Those two groups will go on the 17th, and then the last will be for the last class. Any other questions? I'm going to pass this around. If you guys will just initial by your unis, okay?
And I'll get started. So I'm Julia Nguyen. I spoke last week, so you guys probably met me then. I'm a genetic counselor here at Columbia and have been for about 7 years. And I work primarily with Dr. Chung, who I think you'll meet giving lectures later on. And for those of you who didn't hear last week, genetic counseling it's a master's program after an undergraduate degree specifically in genetic counseling. So you may run into one or two of us throughout your career. So I did things a little bit different. And last week I gave the lecture on pedigrees, and this week I'm going to give you kind of a primer into genetics. And I did that just because of the way your assignments are. So you've had the pedigree introduction, and now we're just going to go into more kind of how genetics has evolved over the years and how it's used today in current practice, what maybe you'll see in your practice and what you will refer for and what indications to refer for. So just kind of a little bit of a background. So just in general, and I think you guys can see this just by, keeping your eyes and ears open to what's in the mass media, is that genetics is no longer an esoteric science that only a few will run into, but it's really something that you will run into on a regular basis as a clinician, and it will impact many people's lives, so in many different ways. So there are genetic factors that account for many things. And I will admit to being slightly biased as a geneticist that we think of a genetic cause more than others, but really there are many different ways that genetics can influence things. And all of these that I have listed on here have a molecular diagnosis that's been identified for at least a portion or a small percentage or a large percentage of things of these particular issues. And so I'll go into more detail, but miscarriages can be caused by a chromosome abnormality. Over 60% of miscarriages, if you do a chromosome analysis on them, you will find a genetic diagnosis for them. Most of them are random sporadic events, which are still important for your patient, because if you can say that this was a sporadic genetic factor, this is then something that you're at no increased risk for to happen again. Neonatal deaths, again, there's a smaller number of those that we can account for as having an underlying genetic cause, but it really is something important for people to be referred to genetics for. So if we can identify a cause, we know what it is. And that's true for all of these things going down the list, is that knowing your genetic cause gives you more power in some cases for treatment, but definitely for understanding risk for other family members. So it's a powerful tool. At the bottom of the list there, actually the drug metabolism is something that you definitely will see as a clinician. So more and more, there are actually at this point, there are already medications that you will prescribe that that the FDA recommends that you do genetic testing for. So warfarin is one of those. So there's the CYP19 metabolism pathway that we all have polymorphisms in, and depending on what our polymorphisms are, we may be a rapid metabolizer or a slow metabolizer. Individuals who are a rapid metabolizer for warfarin need to have, I always flip this around, if you're a rapid metabolizer, you're going to use your medication very quickly, and you need to have a higher dose in order to have effective blood thinning properties, whereas if you're a slow metabolizer, you need to have a smaller dose. If you give them a regular dose of their warfarin, they're going to bleed out. And so this is something that actually everybody gets tested for in some hospitals, and otherwise you do that thing where you give them some warfarin, you have to come back and you test them, you give them a higher or lower dose, depending on how they reacted to their clotting factors. So going back to the beginning, Dr. Gregor Mendel is often considered the grandfather of genetics. He was the man who had his pea plants and identified Mendelian genetics, and he did this by looking at different types of phenotypic, or what the pea plants looked like, and then crossed them and looked at what generations 1 and generations 2 and so on looked like. And so he was uncovering that genetics, or there are discrete factors that influence what something looks like, and that they're passed down onto offspring. And there are traits that can be dominant, and there are traits that are recessive. So here we're looking at classic autosomal recessive inheritance of a dented seed. So you can see that if you have a dented seed crossed with a spherical seed, it is recessive. So in the first generation, all of it is masked, and they're all what we would say today as being carriers of a dented seed. And so when you cross 2 carriers, you have a 1 in 4 chance of having that phenotype, or the dented seed. And so that's just one of his initial experiments that led to understanding genetics. And so from Dr. Mendel, we were able to learn about the law of segregation. So that means that members of each pair of alleles of a gene separate when gametes are produced. That's just exactly what happens when we create our egg and our sperm. We have 2 copies of our gene. 1 copy goes in one egg, 1 copy goes in another egg. So we pass on only 1 copy of each of our genes onto our children. And then law of independent assortment. Just because one particular gene goes in one egg doesn't mean that same gene goes in another egg. So you can have, kind of blonde hair doesn't always travel with blue eyes. You can produce children who have blonde hair and have brown eyes. You can produce children who have blonde hair and blue eyes. You can produce children who have blue eyes and brown hair. The hair color trait doesn't travel with the eye color trait. That's what the law of independent assortment is explaining. There are some exceptions to this rule. If genes are so close together that they don't undergo recombination, or the rate of recombination is very low, there will be certain genetic traits that seem to travel together. So what did he teach us? Exactly like I said, there are discrete factors that determine our hereditary characteristics. Genes appear in pairs, 1 coming from each parent. Genes can occur in different forms, and this is what we're referring to as alleles. You guys have probably been hearing the word allele since 8th grade, and this is what is describing how different genes are different in individuals. And so having a particular allele can give you one eye color versus another eye color, straight hair versus curly hair, the ability to roll your tongue or not roll your tongue. None of these are actually disease-causing mutations. These are just different alleles cause different phenotypic characteristics. So 2 members of each pair segregate during gamete formation, and we've talked about the dominant and recessive. Males and females contribute equally to offspring when we're talking about our nuclear genes. I know last Monday we talked about one particular exception to that, but we're going to go with the basics here. Mendelian genetics is referring to the nuclear genome, so the genome that's in the nucleus of the egg and the sperm, and subsequently in the nucleus of all of our other cells that generate from that progeny. And also their acquired traits are not inherited. So and this is common sense, but believe it or not, it hasn't always been common sense. There were people who thought, so one of the classic experiments of this is if a giraffe stretches their neck very, very long to reach the higher up trees and becomes apt and able at that, then their offspring will have extra long necks, which is the equivalent of saying if I dye my hair red, my offspring will have red hair. And we know that that's not true. But Mendel was actually was the person who proved that that was not true. This is just a nifty little timeline that I like, and you can see how, we were slow going there for a while, and then it almost moved exponentially in terms of our understanding of genetics and our use of it in clinical practice. So if we go back, I believe it starts off in the 1860s was when 1820s, Darwin was doing his experiments, and then we go into in the 1860s when Mendel was doing his experiments. In the 1950s, we first drew DNA, so that was Watson and Crick with the DNA. Same in the 1960s, we understood that there were 46 chromosomes, and then kind of advancing and moving forward, we mapped our first gene, which is the Duchenne muscular dystrophy gene, I believe in the early 90s, in 94, 95, and I might have my dates wrong here because I don't have them memorized and I can't remember them, but in 94, 95 was when the BRCA1 and 2 genes were identified. And when we started patenting genes at that time. If you guys have been listening into the news, the big news in genetics right now is that you no longer can patent our genetic information in its pure form. You can patent at this point cDNA, which for our purposes today we don't need to go into. In 2000, we mapped our first genome, and by 2010, we had mapped the genome of many, many other animals. And so we really have, and at this point, for a relatively cheap cost, you can also have your genome sequenced, compared to the millions of dollars that we spent in the late 90s and 2000s mapping just several genomes. So you guys now all know this, you've been hearing about it for a long time, but Watson and Crick discovered the structure of DNA, along with Rosalind Franklin, who through her microscopy techniques was able to see the double helix, which gave them the tools to truly understand the structure of DNA. And then subsequently, genetics has been making headlines for a long time. So in the 1980s, when we were starting to talk about mapping the human genome, it made the cover. In 1994, we had our first Genetics, the Future Is Now. This is us discovering BRCA1 and 2 and really seeing the tools of genetics applied to the clinicians. We cloned a sheep in 1997. We mapped the human genome in 2002. In 2003, we thought we had our gold standard of the genetic sequence and we could really start to unravel the secrets of it. 2004, we're starting to see individual genes and characteristics and trying to understand more, not just gene by gene, but our genome as a whole, what each individual gene and changes can mean for us. In 2010, this is us getting into kind of the Gattaca world of, does our genetic information predetermine what our life is going to be like, which is not really true. And what does our future hold? As we get into the era of whole genome sequencing or whole exome sequencing, we're able to look at all 20,000 genes at once and learn about things that may not impact us until we're 50 years old, and the likelihood that they will impact us is uncertain. We're really at this point where scientifically we're able to do a lot, but ethically we're still trying to understand what this all means for us and what we should be doing. which is one of the things that I actually work a lot with and I'm very interested in terms of genetics, of really what can we do and what should we do and what is our decision and what is the population's decision. We don't practice paternalistic medicine. So back to the basics, though. You guys all know this, but just a little primer. So we're made up of trillions of cells. Inside each one of those cells in the nucleus are our chromosomes, and our chromosomes are our packages for our genes. So here it is, just all laid out for you. Open up a cell. There are 46 chromosomes in each cell. Unravel those chromosomes, and we have our genes. We have about 20,000 genes. So just some basics about genes and DNA, which you guys have learned it over and over, but just to learn it again. So we know our genes are instructions. They tell our body to grow and develop. They're made up of DNA, and our DNA is made up of 4 base pairs: adenine, guanine, cytosine, thymine, and they pair up specifically. So guanine pairs up with cytosine, and thymine pairs up with adenine, and it's combinations of 3 of these which encode for our genetic code. So combinations of 3 encode for our amino acids. Our amino acids are the building blocks of our protein. So reading our code of our DNA makes these amino acids that are then folded up into proteins and enzymes and create our body and do all the functions of our body. Our code is redundant, so there are multiple sequences that make each of our amino acids. So some of them have 4 different sequences that will make up a specific amino acid, and some of them have 2. And this redundancy really protects us from minor genetic changes that happen will not then change the amino acid that's encoded for there. So I skipped over this slide, but again, something we've all learned. We know that DNA is processed into RNA. The processing of RNA, you read from the coding sequencing of the DNA to create the RNA, and then that's processed into our proteins and our structure and function. This whole processing is very important because we have a lot of DNA that is not encoded into any proteins, and within that DNA there are little structures in there called introns, which are also, they're important in terms of telling us when to start and stop coding DNA, but they're not actually made into protein, and so we need to process that out in our RNA processing. This is a very simple way of saying all of this, and you guys have probably learned it in a more complex way, but for the purposes of a clinician, this is kind of what you need to have in the back of your head to understand the things that you will run into. For the purposes of a laboratory scientist, you need it much more complex than that. So going back to our genes, or our chromosomes which package our genes. So you will hear autosomal inheritance, and what that is referring to are our autosomes. Our autosomes are our first 22 pairs of chromosomes. They're labeled very excitingly, chromosomes 1 through 22, and they're the same in both men and women. So you get 1 copy of each of these chromosomes from your dad and 1 copy from your mom, and it has been that way, and they're the same in a brother and sister, even though they're different genders. They're numbered based on size, so the biggest one is labeled 1, and the smallest one is 22. And these are our autosomes. And genes that occur on these autosomes, when you have a mutation in them, cause autosomal inheritance, so either autosomal recessive or autosomal dominant inheritance. And then we have our sex chromosomes. And as we went over last week, males have an X and a Y, and females have 2 X's. And this is just a little fun fact. Then males, of course, then determine the sex of the baby, because moms just have 2 X's to pass on, so they're always going to pass on an X. And it's whether or not a man passes on a Y in his sperm or an X in his sperm determines whether or not there's a male or female baby. So this is just looking at a metaphase. So you capture the chromosomes at a particular spot in mitosis to get them all long and stretched out, and so you can see the particular banding patterns. And these are specific to each chromosome. So they're not unique to the person, they're specific to the chromosome, and our genes are packaged in the same order and the same location on all individuals, so it's not all mixed around. And this is actually a very good tool for us, because then you know, first of all, this is how we first started to learn where genes are located, by tracking different chromosome abnormalities in different individuals. And secondly, it's very consistent that if you have an extra copy of one particular chromosome, we know what that particular syndrome is, and I think I'll go into that later. And what's most important here is that we have 2 X chromosomes. And believe it or not, this is actually really good resolution. When we first were discovering chromosomes, and even You know, we used to be able to say, okay, there are 46 of them, and we know that there's supposed to be 46 of them. And we really, or we could group them into specific groups, but we didn't know each individual number. So our resolution has gotten better as our science has improved. A male karyotype. I think there is a book called The Shrinking Y, and if you look through evolution, our Y chromosome has gotten smaller for various reasons. I think I show that slide again because we're going into genes. So genes are the sections of our DNA that encode for a trait. So this number is ever changing in the literature, but probably approximately 1.5% of our human genome. So all of our DNA that our body has in each nucleus, about 1.5 is actually encodes for proteins. The other portions are, some people refer to it, some of it is truly information that's kind of it is our regulatory. So it tells us when to turn on and off genes. It tells us in what tissues genes need to be turned on. It's important for replication of our DNA. It's important for processing into DNA and RNA. It's important for repair. And a lot of it just seems to be filler. And actually there's an entire science focused on trying to understand kind of our black box of genetic information and what this means. Because some of it was just important for evolution as placeholders, but we're still trying to figure all of that out. At this point, we think there's about 20,000 different genes. We don't know what each gene is. When I started working, actually when I started kind of learning the field of genetics about 9 years ago, people talked about we must have 100,000 genes. And about 5 years ago, we were working off the number of 50,000 genes. And we were all a little bit dismayed to find out that we probably have about the same number of genes as a mouse, which is 20,000. So we already been over this a gajillion times. We get one copy of each gene from our parents, and alleles are the different base pairs with specific positions in each gene, and changes in them account for variation. So this is just one of the pictures I have. These are all the fun facts kind of alleles. So if you have hanging earlobes versus attached earlobes, when you eat asparagus, whether or not you can smell it in your pee, it has to do with it. When you eat cilantro, if you like the taste of cilantro, or if you think it tastes like soap. In addition to eye color, if you can roll your tongue or not roll your tongue. All different genetic traits that make us uniquely who we are, caused by having different alleles or different base pairs in a specific gene. So this is just going back to nice Mendelian genetics and looking at how we write and how we note this within science. So a genotype is referring to what specifically is going on in your molecular genetics. So if we look at your DNA, what's going on the gene level. Phenotype is what you physically look like. So your genotype predicts your phenotype. There are certain things in your genotype, though, that might mask and you might not present it in your phenotype. So here, this is just, we tend to write the dominant allele or the allele that is visible in your phenotype in large capital letters, and we write the recessive allele or the allele that is masked by the dominant allele in a small letter. So in this particular case, we're looking at the allele for brown eyes, and just so you guys know, we use this all the time, and this is not how eye color really works. It's not a nice two-allele system, but we continue to use it as an example, even knowing that. So having two parents with blue eyes who suddenly have a brown-eyed baby is not necessarily non-paternity, as people might think. So you have a B allele for brown eyes, you have a little B for blue eyes. Someone who has two large Bs, what color eyes are they going to have? Brown, exactly. One with a big B and a little b, what color eyes are they going to have? Brown, exactly. And little b, you're going to have blue eyes in this hypothetical scenario. So we have that, again, just like I said on the last slide and what you guys all knew. Okay. So genetic variations, and this is exactly, so a genetic variation is a difference of an allele in that particular gene. And we all have genetic variation. We need genetic variation in order to evolutionarily survive and also to make us unique to who we are. Genetic variation is a change in the sequence. A variation may or may not cause disease. So something that doesn't cause disease and is frequent within the population, and the technical is probably occurs within greater than 1% of the population, is a polymorphism. So if it doesn't cause disease and occurs within greater than 1% of the population, you have a polymorphism. Versus a mutation is any change in the usual sequence of DNA that causes a gene not to work properly. And so you have a polymorphism versus mutation. I have to tell you that within the field of genetics, there are things that are very clear to us that they're polymorphisms, and there are things that are very clear to us that it's truly a mutation. But as we're getting into the field where we're sequencing more and more genes and we're doing things like whole exome sequencing, there are things called a variant of uncertain significance. What that means is at this point, we don't know if it's a polymorphism, and we don't know if it's truly disease-causing. And some people may say, how can you not know that information? And it's simply because we don't have a large population of people that we've tested. We don't know if this occurs quite frequently in the population. We don't know enough about the genetic change that it's causing. Is it truly disrupting the protein? So we can say definitively, even though we don't know other people with it, we don't know if it's truly causing disease. And so in all fields, this is becoming increasingly a part of practice where we deal with these variants of uncertain significance. And it's actually something that's even within people who are of our non-Northern European ancestry. It's a more frequent instance just because the genomes that we've done out there, we've done more science on people of Northern European ancestry, so we understand their genetic variants better. So how do genetic variants, sorry, I went off on a tangent. But how do genetic variants happen? They're inherited from a parent, and so a parent, either your mother or father, has this genetic variant, and then when they pass on their genetic information, they pass on the copy of the gene with the genetic variant, and you inherit it. The other possible way is that it happens new in you. So at the time of conception, when the egg and the sperm come together, there are little different, so there's all the genetic variants and the genes that you inherit from your mom and your dad, and then there is probably about 10 to 20 new genetic changes that may or may not be true mutations. And this is something that's happening in every single person. So it doesn't have to do with, many people think, well, is it something you're doing during a pregnancy or before you become pregnant or some type of exposure that increases your risk to have some type of new genetic mutation in your child? And at this point, there's nothing that we have seen in terms of exposure for these types of genetic changes in your children. There are some very rare instances of things where there's a particular exposure to a medication that increases your risk for children with particular new genetic mistakes. But for the vast majority of these genetic changes that occur in us, it's not something that's anything was caused, but rather it's just chance. There are also new genetic changes that can happen in you after you're born. These are referred to as somatic mutations, and I think my next slide, I go into that more. So I'm going to come back to somatic mutations, but we'll get there. So germline mutations are the mutations that are inherited from your mother or father. Germline means that they're in your parent's egg and the sperm, and then they're subsequently in every single other part of your body, because the child is generated from the egg and the sperm come together, and every subsequent cell that you have is generated from that egg and the sperm, and so all of your cells in your body have that particular mutation. This is why you can take a blood sample and learn about your genetic information for your heart, for your brain, for your cancer, because your blood cells have the same genetic mutations as every other cell in your body. This is the same also for those new de novo genetic changes. Those are still germ-line mutations because they occurred so early at the time of conception. Every single one of your cells has them, or nearly every single one of your cells. New mutations. Which I just said. So somatic mutations are mutations present in other cells of the body, but not in the germ or reproductive cells. And they're typically isolated to one area or one particular tissue. And these are things like, we talk about having, genetic mistakes causing cancer, but yet the cancer is not an inherited form. So in all of our cancers, in that particular tumor, if you look at it, there are going to be multiple genetic mistakes. And those are somatic genetic mistakes, meaning that if we took a blood sample, we wouldn't see those genetic mistakes. But if you take a sample from the tumor, you're going to see multiple genetic mistakes because something has happened to the regulation of those cells. They have not learned to die when they're supposed to. They're generating in mass to create a tumor, and it's because the genetics in them is wrong. And these particular genetic mistakes are things that happen in all of us, but fortunately we have the tools to correct and repair. So you guys have all at some point probably learned about looking at different types of repair mechanisms for our DNA. So we have things that actually excise out a base pair when it's incorrect, or we can pull out a whole chunk, or we can replace the base pair. So you talk about double-stranded DNA repair, and different types of repair mechanisms which thankfully save us from most of the consequences of having DNA damage. But in some cases, eventually our ability to repair these can fail. And so you see cancer, you see aging, you see other types of disease processes And these are all caused by somatic or new mutations. Somatic mutations are not passed on to your children because they're not in your germline. Okay. So going on to kind of what does a genetic mutation look like? So here we're looking at the beta hemoglobin gene, which is on chromosome 11. We know it's at the tip. And if you unravel that chromosome, you would see a particular nucleotide sequence. So you're seeing CTG, so each of these base pairs. And these, remember, we said each of those base pairs encodes for a particular amino acid. And if the amino acid is incorrect, then our protein isn't made correctly, and we have a genetic disease. So in this particular case, you have a GAG, which encodes for glutamic acid in your normal beta hemoglobin. And so you have normal red blood cells. Whereas if in your beta hemoglobin, and this needs both copies because sickle cell disease is an autosomal recessive trait, but if in both copies you have a GTG, which encodes for valine, a different size, different shape amino acid, it causes a mutant protein, and subsequently the phenotype of your red blood cells are the sickle shape. They don't transport oxygen to the parts of your body as well, and so you have the hemolytic disease of sickle cell anemia. And so that's just kind of the whole process from chromosomes and DNA down to encoding for the protein. Okay. Most of these are things that we've covered already, but it's just kind of a list and summary of things that we, to kind of run down everything from the lecture here on. So genes interact together to form proteins. So there are some proteins that are encoded by one single gene, and then there are other proteins that they're complex and are encoded by multiple genes. So if you think of our red blood cells and the hemoglobin that carries, we have both beta and alpha hemoglobin. So we have multiple genes that encode for our hemoglobin. Expression varies among cells, even within the same tissue. That's just common sense. Of course, the cells that are expressing genes in our blood cells, the genes that are expressed in our blood cells are different than the genes that are expressed in our brain cells, in our skin cells, in our heart cells. While we have the same complete copy of our genetic information in all of our cells, different genes are turned on in order to make our cells specific. And in fact, for these cells, the other genes are turned on and silent to the point where you cannot turn them on again. That's the whole basis behind stem cell research, is that I can't turn a skin cell very easily into a heart cell and so on. We wish we could, because it would be nice to be able to take an easy sample and turn something into a cell that we need to be regenerated for a disease. For the central dogma of DNA to RNA to protein is true. There are some cases where that doesn't always follow that particular pathway. We did not cover this, and I'll go into it in more detail, but we talked about how there are regions of the DNA that are not encoded for protein. In particular, when we look at a particular sequence of DNA that's encoding for a gene, there are both introns and exons, which the introns are the placeholders and the exons are actually encoded into RNA, and the RNA is then read for a protein. Genes can encode for many proteins, and so this is because of the way we have our regulatory regions, we can make different proteins out of the same sequence of genetic information by starting in one region versus stopping in another, or encoding for one portion but not another. And that's going on to, again, genes can overlap or nest. This, I'm not going to touch a lot about imprinting, but imprinting is a phenomenon by which, depending on whether or not you inherited the gene from your dad or you inherited the gene from your mom, whether or not you express that gene. So this is not genes on the X or the Y chromosome, but it's rather genes that are on the autosomes, so that you get copies from mom and dad. But whether or not you're expressing that particular gene impacts whether or not you're expressing that particular gene is based on whether or not you receive that gene from your dad or you receive that gene from your mom. So there's paternal imprinting and there's maternal imprinting. And not something that you need in your day-to-day life. There are many geneticists who don't understand it. And we certainly don't understand the mechanism completely. So genes can be unstable and change in size when transmitted. So there are particular genes that have these triplicate repeat regions, and this is something, so within our amino acids, if you had a triplicate repeat is referring to the fact that you have 3 base pairs and it repeats itself over and over. And there are particular genes where this happens, or there's a palindrome, and all of those where you have repetitive sequencing is more susceptible to having a particular mutation or something happen to it, because that is when you can have overlap or mismatch of your reading frames and causing the gene not to be read or encoded properly. And I'll go into more detail about that triplicate repeat. Okay. And there's just a lot we don't know. So genotype, we went over that again already, that there's very small variability in individuals. And let's get into how we look at our genetic information. So we have kind of an overview. We understand what genes are. We understand what alleles are. We understand what inheritance is. And so now that we understand all of this, how can we actually look at this information in clinical practice? And what do we as geneticists and genetic counselors do to try and understand what's going on in an individual's genetics to try and aid in how we care for them? So the most basic and the thing that's been around for about 30 years, it's definitely improved over the past 30 years, is chromosome analysis and karyotyping. So being able to look at our chromosomes. Before, geneticists were doing things like mapping our hand lines, literally, and this was geneticists were doing this. And there were other things doing a lot of different measurements of facial features and symmetry and things like that. Before we were able to we knew that there were differences about people's physical features that were caused by genes, but we didn't have the ability to look at their genetic information on a molecular basis. Chromosome analysis and karyotype are used, they're synonymous with each other. Over the past about 8 years, we've been doing a technology called a DNA microarray. And this is something where we're able to look at the chromosomes in more detail than just looking at that banding pattern, and I'll go into more detail with that. And in between chromosome analysis and microarray, we were doing something called FISH, or fluorescence in situ hybridization. And this is something where it's literally just saying we tag something or we mark something with a particular color when we're looking for a particular region. When you're doing a fluorescent in situ hybridization, you need to know specifically what you're looking for. So I'm looking for a deletion on chromosome 22, or I'm looking for a deletion on chromosome 7. You can't just go into it willy-nilly and say, you need to have a particular condition in mind to do a FISH, whereas a microarray will look simultaneously for all different small deletions and duplications across all the chromosomes. So a microarray has pretty much replaced FISH. And then we have single gene analysis, which is gene sequencing, and more we're using exome sequencing and genome sequencing. And I'll go through each one of these individually. This is one of the analogies that is often used to explain how analyses are done to a patient. So the most basic way to think about our chromosome analysis is if we think of our 20,000 genes as a set of encyclopedias. When we do a chromosome analysis, we're simply looking to make sure that each book is there. So we're saying, is there any missing or extra books? So this by no means is getting to the nitty-gritty. For all we know, when we do a chromosome analysis, if we open up one of those books, it's completely blank or something like that. And so this is something where we're not getting to a very detailed genetic basis, but we're definitely getting the gross picture. And this is something that's very important because we know of particular conditions that are caused by having whole, missing, or extra chromosomes. But it certainly does not answer all of the genetic cause or question when we're doing testing. So having a normal chromosome or karyotype result does not mean that you don't have a genetic condition. So chromosome disorders. So having an extra is trisomy, having a missing is monosomy, and they're caused by mistakes in meiosis, or the separation of chromosomes in the sex cells and the gametes. And one of the things we learn about, and I'll go into more detail, is the risk to have a trisomy or have a chromosome abnormality in a pregnancy increases with maternal age. And so everybody's been exposed to the fact of things like advanced maternal age, or the fact that your risk to have a baby with Down syndrome increases as we get older. I didn't bring those charts with me. I usually try to. But this is just looking at exactly how aneuploidy, or having the wrong number, aploidy is exactly what I'm saying, aneuploidy, occurs. And it occurs by having the wrong number of chromosomes in your egg and your sperm. So in this case here, egg and sperm have nice 23 chromosomes and 23 chromosomes, so you make a nice egg with 46 chromosomes. You have 2 copies, or 2N, of each of your chromosomes. Whereas if you have an extra copy in the egg, or you have a missing copy, but in this particular scenario, we look at an extra copy in the egg, you then end up with 2N plus 1. So one of your copies of your chromosomes you have an extra copy of. The most common trisomy is trisomy 21. Does anybody know what trisomy 21 is? Down syndrome, exactly. Having an entire extra copy of chromosome 21 is Down syndrome, and almost everybody out there with Down syndrome has this particular genetic mistake. There are a small number where they have an extra copy of 21, but it's jot to another chromosome. So they actually only have, when you count them, 46 chromosomes, and that's called a Robertsonian translocation or translocation Down syndrome. But the vast majority of individuals with Down syndrome have it, and it looks just like this. And it was caused by non-disjunction in the maternal gametes. So somewhere either in meiosis 1 or meiosis 2, there was abnormal segregation of the chromosomes, and you resulted in an egg that had 24 chromosomes rather than 23. The risk of that increases with age. So I think at around 22, it's like 1 in 5,000, or that's wrong. It's more like 1 in 500. When you get to age 35, it is 1 in 200. By the time you get to age 47, I believe it's 1 in 12. So just to kind of look at how that changes over time. That also means, though, but if you think about 1 in 12, that's still less than a 10% chance of it happening. So it's all about risk perception. So this is Down syndrome. You guys have all seen someone with Down syndrome. And just as I said, the majority of them are caused by maternal meiosis 1 error. Everybody has some degree of mental retardation. That does not mean they don't have the capacity to learn and actually to become quite independent. Although most individuals with Down syndrome will not live independently, will live in group home settings or with relatives. But there is, even within Down syndrome, with all genetic conditions, there is a spectrum of severity. And so there are kids with Down syndrome who are high functioning, there are kids with Down syndrome who are low functioning. And for many kids with Down syndrome, ages 1 through 4 really don't look that much differently than other kids. They have higher risks they have higher problems with hypotonia and feeding disorders, and they do have somewhat delayed in meeting their milestones, but caring for them is very similar to young children. And where kind of the real separation happens is as they become in adulthood, and kids of average mental capacity are gaining more independence. There are those significant birth defects that occur with them. Nearly half of the kids have a heart defect. 10% have duodenal atresia. There can be clefts and cataracts. They have shortened life expectancy. And mortality is quite high in the first year of life because the kids with the more severe birth defects associated with their Down syndrome pass away from them. They also have an increased risk for thyroid disease and leukemia, and as we're learning, dementia at a younger age as well. So one of the other more common chromosomal abnormalities is trisomy 18 or Edwards syndrome. So this is, again, something that we can pick up prenatally, and it's something that we see often with birth defects on the ultrasound because these kids are more severely affected. And kind of the layman's way of understanding of that is that chromosome 18 is a lot bigger than chromosome 21. There's a lot more genes, and there's a lot more influence in our developmental. So this is a kid that's more severely affected with trisomy 18. Many of these kids don't make it to term and can result in miscarriage. And children who do make it to term don't do very well. And this is because of their neurocognitive problems as well as a heart defect are some of the things you can see. So this kid here, when we look at this baby, you can see microcephaly. You can see micrognathia, which is the chin is recessed. You can see abnormal clenching of the hands. They often have overlapping of their digits. And the feet have both contractures as well as rocker bottom feet, as well as abnormality of the toes and overlapping toes. And these are all features. Kids with trisomy 18 don't always look this severe, and they actually can be very pretty babies in terms of just little petite kids that don't look that sick. But unfortunately, neurodevelopment, they do things like apnea because their central brain is just not working to tell that baby when to breathe. And it can be a difficult situation in terms of if a baby is born alive and the baby doesn't have a heart defect, it becomes difficult to try to figure out ethically how to treat that baby. There are kids, though, that definitely survive past a year of age and much longer. Many of them don't gain very many milestones, and that they are severely neurocognitively they have issues, as well as they have things like need breathing assistance, they need feeding assistance, they need 24-hour care, they need significant care. And for many of those babies, they are probably mosaic trisomy 18, meaning not all the cells of their body have an extra copy of chromosome 18. And so that's why their severity allows them to get past the neonatal period. And I had more things on there. Anyway. So trisomy 13, this is an extra copy of chromosome 13. Trisomy 21, 18, and 13 are the most common live-born chromosome abnormalities. The other chromosome abnormalities occur, but they don't result in live births, and many of them don't result in recognized pregnancies because of the degree of abnormality. They just are not viable. So trisomy 13, also called Plateau syndrome. This is the one that we think of midline defects. So brain, clefting, heart, feet, polydactyly as well. So you see cardiac defects, you see whole and prosencephaly, you see midline cleft lip and palate microcephaly, cutis aplasia, which is the lack of the skin. It can also just be a lack of hair, but there you're seeing lack of skin. It can go all the way to the skull. Polydactyly, and it's post-axial polydactyly, so this is on the outside, an extra finger or toe on the outside there. You can have small eyes, or anophthalmia, which is where you're missing a eye. You can have coloboma, which is where there's that slit or cutout from some level of your eye, either in your iris or your retina or your lens going back, and low-set ears with abnormal chelaces, so they don't have as many folds in their ears as we do. With all of the conditions that I've talked about so far, it's not that you have every single feature, it's that you have some combination of the constellation of features. With both trisomy 18 and 13, there is an over 80% and probably higher than 90% chance that on your level 2 ultrasound, which you get at around 20 weeks of pregnancy, we're going to pick up some abnormality that gives us some idea that this pregnancy might be affected with it. And then you can talk about testing options. I believe there's another counselor coming in, though, to talk in more detail about the testing we can do during a pregnancy. The other common chromosomal abnormality that occurs in liveborns is Turner syndrome, or 45X. So you have monosomy X in this particular case. These are little girls for the majority. They have the heart defect that's most commonly associated with this condition is coarctation of the aorta. So it's actually a portion of the aorta in your arch is actually constricted in some way, and so you have abnormal outflow. You also see webbing of the neck, a shielded chest or wide-spaced nipple, lymphedema. Majority have structural abnormalities, short stature, and infertility because you result in never developing of your ovaries. You get what's called I'm totally blanking on it. One of the things with Turner syndrome is that oftentimes you have some other cell line or some other chromosome combination with it. So you can see kids where they have 46X in some of their cells, they have 46XX in their other cells. You can see kids with 46, and those they can have different severities or degree of the spectrum of their symptoms. One of the things we're not good at, and we can't predict, though, is what degree of severity. So some people, we work a lot at trying to understand, if we take a blood cell, what percentage of those blood cells have a 45X line, and what percentage of those blood cells have a 46XX cell line, and does that predict the particular phenotype or the severity of Turner syndrome that we might see in that child? And it really doesn't, because you know what it looks like in the blood, but I have no idea what the level of the mosaicism is in the brain, is in the ovaries, is in all the other tissues of the individual that is affected by this condition. You can also see Turner syndrome where you have 45X, 45XY. These are typically phenotypically female babies, and it's that they've lost a Y, and they developed a phenotypically female. You can also have things called ring chromosomes, where one of the X chromosomes has lost portions of its telomeres and then looped around to make a ring. So one of the things that's important with Turner syndrome is just is the fact that there are varying molecular diagnoses, and it is important for them to be followed by a geneticist. And for some of those kids who have the mosaicism, if they have a significant portion of their cell line that is 46XX, there are things, treatments that are looking into ovary preservation. So if we can somehow remove and save ovaries before they go into I can't remember the term at this point, I'm sorry guys, before they fail, before they shut down and fail and have an increased risk for malignancy, there's some thought of preserving fertility in these kids. It's annoying about that. So, I'm still blanking. So going into the next section, we're looking at chromosomes. We talked about looking at structural abnormalities in chromosomes and having missing or extra chromosomes. But if you've got a kid that still seems to have some type of underlying genetic condition and you have not found it in your karyotype analysis, the next step is a microarray analysis or chromosome microarray deletions and duplications. So the analogy I'm using here is you've pulled your books off your shelf, you're opening them up, and you're looking at each page to see whether or not the page that is missing or extra. So you're not reading each gene, you're not reading each page individually or sequencing each gene. Rather, you're looking to see, are there missing or extra pages? So it looks to see whether or not there are missing or extra pieces of whole areas of your chromosomes or of your genes. So it looks at probably about the 10 to 100 gene level. This is showing a computer readout where they've done a microarray analysis, and this is just a cartoon of the chromosome, and this is looking at the copy number. So how many copy numbers of a particular region of the chromosome does this person have? Okay. As I was saying to you before, that fluorescent in situ hybridization was the old way of looking for microdeletions and duplications. The downside of this is you had to know what particular chromosome abnormality you were looking for, so you knew what particular hybridization you needed, and so it was great for those that we are aware of and those that have specific phenotypic features, but it's not the most efficient way of doing this testing anymore. So in this particular case, again, just a computer readout that's showing kind of what things look like. So in this particular case, when you have all the normal amount, your copy is 2. When you have a deletion, you have only 1 copy, and when you have a duplication, you have 3 copies. Okay. This is looking at common microdeletions and duplication conditions, and these were in the past diagnosed by FISH and more recently are diagnosed by microarray. One of the ones that if you work in pediatrics you will likely run into a child with DeGeorge. This is 22q11.2 deletion. It's also called velocardiofacial syndrome. And deletion 22. And these kids, I'll go over to the next slide. These kids do relatively well, although many of them have congenital heart defects. The rest of these conditions are actually more severe. Miller-Dieker and Smith-Magenis are not very common, and these kids are severely affected. Prader-Willi, this is the one where you often hear about it because this is the condition where you have an insatiable appetite. So these kids often become quite large. They have and they have feeding issues and the fact that they're food seekers. So people have to, they'll dig through garbage cans. They will open up padlocks on your refrigerator to get to food because they just don't have the cessation of being full. Fortunately, we've found recently that actually growth hormone therapy can help these kids significantly with that particular issue. Angelman syndrome. So Prader-Willi and Angelman syndrome are in the same region of chromosome 15, and it has to do with whether or not the gene that you inherited from your father versus the gene that you inherited from your mother is affected. And this is one of these genes that I talked about as having imprinting. So whether or not you receive it from your mom or your dad impacts what particular condition you have. Angelman syndrome has more severe, typically more severe neurocognitive issues. It's sometimes called the happy puppet syndrome. Kids tend to have a characteristic flapping. They have severe ataxia and oftentimes have no speech. Williams syndrome is a deletion on chromosome 7, q11.2. There was a woman with Williams syndrome that was frequently on 20/20 for a while, and she really liked playing. She liked music and playing the piano. These kids, again, still have no cognitive issues. They sometimes have a heart defect called supravalvular aortic stenosis. So right above the aortic valve, they have stenosis of the aorta, which can cause problems, and they may need ballooning or other treatment for that. And the other characteristics of these kids is they actually are very sociable, and so they can be missed for a while because they're so loquacious and interactive that it kind of masks some of their neurocognitive issues that they have. Many of them have a great love for people and for music, for objects that spin, and one of the things is they also don't understand kind of danger, so they'll hug anyone and everyone. It's one of the stereotypes of them, and also one of the features. But again, all of these have variable levels of severity and variable features in each child. Krita Shah is actually named for these children have their their cry is like a high-pitched meow, and so this is back when we got really inventive with names, and so Krita Shah. And these kids are severely neurologically devastated. And then Wolf Hirschhorn's is one that's actually sometimes called the Greek helmet face. They have a flattened face and flattened nose and flattened features, and have one of their biggest issues is significant feeding problems early on. So here's DeGeorge syndrome. This is one of the most common deletion duplication syndromes. It accounts for a large proportion of congenital heart defects, and every kid in our NICU with a congenital heart defect gets tested for DeGeorge. There are particular heart defects that are more common in DeGeorge. They're thought of as conal truncal defects. So these are the portions, if you think of your embryology course of the conal truncal portion of the heart, so you think of tetralogy of Fallot, the outflow tracts, these are the things that are impacted in DeGeorge syndrome. You can also have thymus issues, so you can have hypoplasia of the thymus or the parathyroid, facial dysmorphism, so we most often think of a bulbous nose. You can have a flattened upper lip and low-set posteriorly rotated ears. Many of these kids can look okay. They can have palate issues. It can be most severely as a cleft palate or a cleft lip, and most minor as a soft palate abnormality, which you may not catch right away, and it might be something that only becomes evident when they're talking, in which case when they often have a high kind of nasal-pitched speech. They can have renal structural abnormalities, and they have learning disabilities. And one of the things that occurs in these kids is actually psychiatric disorders. So there are some people where you diagnose it when they are diagnosed with schizophrenia, and you go back and you say, well, they also had some learning problems, and yeah, they always had a high-pitched nasal voice, and they got a lot of urinary tract infections, and while they never had the heart defect, because not every kid with DeGeorge has a heart defect, you diagnose it at that point. One of the things, though, is that the reason why we're so quick to do this test is because early diagnosis really does help with these things. If you know that a child is at risk for learning disabilities and psychiatric disease, then you're going to implement interventions a lot quicker and hopefully have a much better outcome for these kids. As well as know, I need to look at those kidneys. If this kid only has one kidney, I need to know we need to protect that kidney and be very careful about it. A hypernasal speech in the cleft palate, that's something that can be repaired surgically and improve things like feeding as well as speech. They can also have hearing loss. So moving on, we've covered how to pick up large chromosomal abnormalities like Down syndrome with a karyotype. We've picked up deletions and duplications that can cause a myriad of birth defects as well as developmental issues, and now we're looking into single gene mutations. So this is in our 20,000 genes, looking for particular mutations that are causing a particular disease. And how do we do this? The way I like to think of it now is you have your set of encyclopedias, you've made sure that all the pages are there, and now you're looking at each page individually. And at this point, we're moving towards a complete scan of every single page and every single word, but there's also at this point we tend to look at a specific gene that we know is implicated in that particular condition, and we sequence or we do single gene analysis of that gene first or that family of genes that's associated with that particular condition. So this is looking at a Sanger sequencing, so there's a different peak for each base pair that's occurring there. It's a different color, and you expect to have one peak when there's the same base pair present on each strand, or the same allele on each strand, and you expect to see two different peaks when there's two different alleles present, when there's an A on one strand and a T on the other. And then there's a particular sequence that you expect to see versus what you see in the patient. So this is just very basically how, not very basically, but this is how they do Sanger sequencing. Each particular base pair is labeled with a different color, and you give them a primer, and it goes and goes and goes until it doesn't have what it needs to create that particular sequence, and it creates a size of a band that's then read out on a gel. So single gene mutations cause our Mendelian conditions. So full circle here, back to all those conditions we talked about in terms of their inherited autosomal recessive, dominant, or X-linked excellent fashion. These are conditions that are all caused by a single gene mutation or having one specific allele that is a mutation on that particular gene or in both copies of your gene if you're dealing with recessive conditions. So I think we talked about most of these last week. Some of the most common autosomal recessive conditions that we talk about are cystic fibrosis, which is a lung disease, spinal muscular atrophy, which is a muscular dystrophy. Autosomal dominant conditions are things like Marfan syndrome, which is that arachnodactyly or very long arms and legs with the issue with the aortic dissection or aneurysm. We talked about most of the cancer conditions are autosomal dominant. Some more common X-linked recessive conditions is hemophilia. X-linked dominant, not as common of a particular inheritance pattern, but one of the conditions there is incontinentia pigmentae. So when we're looking for these particular conditions here, we do this by just doing one specific gene analysis. We know of one specific gene, the CFTR gene, that we all have 2 copies of it, and when you have a mutation in both copies of this gene causes cystic fibrosis. This has it's monogenic inheritance, and it's homogeneous in how it's actually the genetic cause of it. In that it's very clear to us with cystic fibrosis what the genetic cause is. Whereas with other genetic conditions, we know it's genetic, but we don't know what the specific gene is, or there's a whole family of genes that cause it, in which case doing single gene analysis is not the easiest thing to do. So for example, retinitis pigmentosa, which is a particular problem with the retina that causes, over time, causes blindness, there are over 20 genes that are associated with that. And so it costs a lot of money and it takes a lot of time to do single gene analysis of each one of those genes. And so until now or until recently, it's been very difficult to do genetic testing for that and come up with an answer. And so this is where the new technology has come in. So very briefly, I'll go over again. This is going into now looking at whole exome and whole genome analysis. And first of all, is whole exome analysis. So remember we talked about a particular gene is composed of both coding regions and non-coding regions. And you go from DNA to RNA to your protein sequence. And when you do that process, you read just the regions that you need to make your protein. The other regions are important there for placeholders and regulation. But what you can see here is that there is a gene, and there's the introns, which are not read into the protein, and there's the exons, or the yellow sequencing, that are spliced out and read to make the protein synthesis. So you go from DNA to RNA, you take out your introns, and you encode for your protein with your exons. And so what we now have the technology to do is essentially take this type or that sequence and look just at the exons of all of our genes across our entire genome. The thought process behind that is that the vast majority of disease-causing mutations occur within the exons of the genes and not in the introns. And this is based on a lot of research of looking at all of the genes that we know of and all the mutations that we know of. There are certainly mutations that occur within our introns, but to do a fast and quick, dirty way of doing testing, and the cheapest way to do this to get our answer as fast as possible is to look at the exons first. We think about 80% of our genetic mutations are present in our exons. And so it's essentially like selective reading. I think you guys have all seen this passage where your brain can kind of read through that to read what it wants to, somewhat. And you can take it and you can see, so we've taken out all the junk in between and looked just at the exons so you can now read 407 years ago. So that's what, it's this, if you think of this entire passage as our genome, our exome is what's in yellow, what's important. This is a very complex schematic of how we do exome sequencing. It's chip technology, if you ever are running into that, and we talk about SNP analysis. When is whole exome sequencing used? So it's used when a genetic syndrome is suspected but a gene is unknown. We call this the genetic odyssey. So a family has been to us, and most of these families have had some type of genetic testing with us. They most definitely have all had a chromosome analysis and a karyotype, and we still have not identified the testing. And a genetic cause is suspected in these families, one, because there's a family history of the particular condition that the person has, or 2, because that child has syndromic-like issues. So it's that child has both a heart defect as well as learning disabilities, as well as possibly neurocognitive muscular problems or hearing problems. They have multiple systems involved. So a particular, it can't be, the most logical explanation is there's one gene involved in all of those systems that has a mutation in it that's causing the disease. The second process that we use it in is where a condition is very is genetically heterogeneous, meaning that there are multiple genes that cause that one particular condition. So I use the condition retinitis pigmentosa, but there are other conditions where they have multiple genes that cause one particular condition, and there's no definitive phenotype or kind of allows you to particularly pick what gene is. So another condition, there are inherited arrhythmia conditions that are genetically heterogeneous, and there's nothing about their EKG or their symptoms they have that target me towards one particular gene. And so because I can't pick, and I'd have to go through each gene individually, the best way to do it is to simultaneously test all of the genes at once. And finally, this is increasingly something that's going to be used for personal genomics. So you're going to have healthy individuals who are getting their exome and their genome sequenced to understand, one, about their genetic mutations that they may harbor and cause them to have disease later on in life, as well as about their polymorphisms that may incur a relative risk or an increase or a decrease for particular multifactorial traits. So there are certain polymorphisms that we know slightly increase your risk for age-related macular degeneration. And some people, I want to know that so I can take precautions, or slightly increase your risk for Alzheimer's or your risk for hypertension or diabetes. And this is something where the science is still really new. For all of the things that are out there that you can easily do this type of personal genomics, it's kind of a gimmick and a hoax at this point, but I think this is something that will increasingly become reality. So just going into more kind of where this really is clinically useful in terms of personal genomics is drug metabolism. So I alluded towards this. There are already medications that are well understood that having particular polymorphisms impact the rate at which you process them and impact the dosage of the medication. The other place that this is coming very much in is playing a role is in developing chemotherapy. So we already know about ER positive or ER negative breast cancer and whether or not there's particular chemotherapies that respond well to that, or tamoxifen after you have your breast cancer. And we're learning that each person's tumor is genetically unique, and knowing genetically what your tumor is made of can help to target your chemotherapy better to that particular cancer, to that particular cell type that has a cancer, rather than chemotherapies that are killing both your good cells and your bad cells. That's what causes all the bad side effects of chemotherapy in the most general sense, is because you're having your good cells killed along with your tumor cells. So targeting that is one of the things that whole exome sequencing of your tumor is going to lead to. The other thing is, this is very much what you see in kind of the direct-to-consumer tests that you see online, like 23andMe or Ancestry.com. These are really used more, I don't know. It's not something that's going to impact your health care, but definitely something that people are very interested in, which is Ancestry. So understanding kind of, am I a long-lost ancestor of Genghis Khan, and those types of fun things. And then drug design, just like I was talking about before. So I think this is where genetics is moving towards, and this is something that I think will become very much part of clinical practice. And these are the things where we're seeing it. I think drug metabolism and drug design are what's going to happen first, and maybe at some point we're going to figure out this personal genomics to understand our disease risk and possibly impact how we live our lives. But studies have been done looking at people who have been told they have you have a one-fold increased risk for prostate cancer compared to anybody else. Has that changed how they've gotten screening for their prostate, or the foods that they're eating that we think maybe increases their risk for prostate cancer? For the most part, people don't change their health habits based on these small changes in the relative risk that are incurred by particular polymorphisms. And then, like I was said, I think earlier in the lecture I alluded to this, is that the vast majority of us in clinical genetics are focusing on those things that we understand, and thankfully we have a lot of people out there in bench and research genetics that is saying, hold up a moment before you get all excited about everything else that's happening and ignore this other half of it. We need to take a look at this again. I can't quite believe that 97% of our genome is what you call junk DNA. And we really are learning that this is not junk DNA. This is really important for the regulation and processing of our genetic information. And so I think there's a lot more to come from this seemingly dark matter genetic information, and there's a lot of work that's going on out there. So genetic-based medicine. I've touched on this a little bit. Why do we do genetic testing? For many people, we do genetic testing to clarify the diagnosis. We need to know the diagnosis. What does this person have? Is this truly a hereditary breast and ovarian cancer, in which case this individual needs to talk about other prophylactic treatment for their other increased cancer risk? Is this truly Marfan syndrome, in which case I need to monitor their eyes very closely for lens dislocation? So having a particular phenotype or features of a condition, you then have genetic testing to figure out your particular genotype, which then can have implication for your treatment or your surveillance. There are some cases, though, where genetic testing is not going to impact your treatment or surveillance in any way, but it's really something to give you closure on. So if you have a child who's had significant birth defects, neurocognitive issues, knowing your genetic test result in many cases today doesn't immediately impact their treatment, but rather tells their parent what this child has, as well as impacts what's going to happen in the future in terms of what's the risk for other family members. So clarifying, there are some cases, so in addition to clarifying your diagnosis, it can clarify prognosis, so for how aggressive a tumor could be. Aid in prevention, so exactly like I was saying, if you have a hereditary cancer syndrome, you know you're at risk for other cancers, and how can you go through screening or have prophylactic treatment. There are some genetic conditions in which there is treatment in place, and there's really good treatment, and there's treatment that pretty essentially cures you of that particular condition. And these most commonly are metabolic conditions. So unfortunately, the vast majority of metabolic conditions, there is no treatment, but there are a subset of metabolic conditions that do have treatment. So this is one of these things that we very aggressively, if we have any type of suspicion that the child has a metabolic condition, we do very comprehensive screening of it through looking at blood and urine samples to look at how they're metabolizing processes are happening. Because things like Fabry's disease and Gaucher disease and Pompe, and all of these are lovely doctors who name conditions after themselves, have enzyme replacement therapy, where we actually give back the enzyme that that person is missing or isn't making because of their genetic defect, and essentially either prevent the progression of the condition or reverse some of the symptoms of the condition. And because we have these therapies, that's why it's so important to make those diagnoses. Gene therapy. Pretty much everybody who gets a genetic diagnosis then asks about gene therapy. We really are in the early stages of gene therapy, and I've been saying that for the last 10 years. It feels like it's a slow progression. The process of going back in and fixing your damaged gene that is present in every single one of your cells is really difficult. These are things where you learn about kind of viral vectors where you try to insert that gene, but you have to get that gene back in the correct spot. You can't disrupt any of your other genes. And so gene therapy, we are definitely working on it, but it's difficult. And then drug therapy. I alluded towards this in cancers, that if we know specifically what the genetic cause is of your cancer or what your tumor genetically looks like, there are particular drugs that work better than in others. And we know this best in things like colon cancer and breast cancer, but I think more and more it's becoming other things. And I've talked your ear off about personal genomics already. So just some kind of fun things. So myozyme for Pompe. This is a rare metabolic condition in which a child does not produce something called acid maltase. And for these children, they're just fine. And subsequently what happens after a couple months of age is actually several weeks of age, you notice severe hypotonia. So they're just completely floppy babies. And then if you look at their heart has cardiomyopathy or thickening of it. And what that's happening there is they're not processing their energy properly. And they're having a buildup of this particular glycogen storage disease. And without this particular enzyme, they're not getting rid of their waste material properly. And a couple years ago, this movie was made actually about one father who had 2 children with Pompe disease, who really drove science to spend money on trying to come up with a enzyme replacement therapy for this. And this is actually, it's an amazing thing to watch, because kids that would normally end up completely hypotonia, no movement, never gain a milestone, eventually be trached, eventually pass away from this condition, now are doing incredibly well. There are still generally some myopathy, degree of myopathy that they have, but they're walking, talking, running around, kicking a ball, eating on their own, not having cardiomyopathy because of this enzyme replacement therapy. It's not the case for every kid that we use this on, but it is really pretty impressive. And there's actually a doctor here at Columbia, Dr. Slonim, who was involved in identifying Myozyme for Pompe. So. Gene therapy. So again, this is, I touched briefly on this. So the ones that are moving the most towards having possibly some effect with gene therapy, one is Duchenne muscular dystrophy. And this is only a small subset of children who have Duchenne muscular dystrophy have a particular stop codon. So what happens is their protein is truncated. They have a codon that's, rather than encoding for a particular amino acid, encodes for a stop codon, in which case they stop the translation into protein at that particular spot. And what this gene therapy is focused on is actually reading through that stop codon. So if we can skip over that stop codon and then just keep reading all the amino acids moving on, or excuse me, all the base pairs to encode for amino acids moving on, you then can create a protein. You can create your protein, and it's slightly, your dystrophin protein is a little bit different, but you now have more dystrophin. So the problem with Duchenne muscular dystrophy is you have decreased levels of dystrophin, which is important for muscle structure, and what they're doing with this particular is they're reading through to create more dystrophin and hopefully reverse some of or prevent some of the symptoms of Duchenne muscular dystrophy. Spinal muscular atrophy, again, in this case, you actually have a pseudogene that the structure of the gene and the protein that's made is very similar to the protein that's altered in spinal muscular atrophy. So you have SMN1, which when you have a mutation in SMN1 causes spinal muscular atrophy. But everybody also has SMN2. And some people have more copies of SMN2. And so the idea behind, the SMN2 protein is very similar to the SMN1 protein. And the more copies of the SMN2 protein you have, even if you have a mutation in the SMN1 protein, the less severe your symptoms of spinal muscular atrophy are. And so based on that knowledge, that if you have more SMN2, you have decreased severity, they thought, what if we could increase the expression of the SMN2 that these individuals have? And so that's what this therapy is focused on, is increasing the expression of a pseudogene, or a gene that encodes for a protein that looks almost like the protein that you're missing. Okay. So, an overview. What have we learned? We've reviewed that cells have chromosomes have genes, and our genes are made up of DNA. We know the 22 pairs, we have about 20,000 genes, and having missing or extra copies of your chromosomes can cause genetic disease. We know about 1.5% of the human genome encodes for protein genes, and the DNA encodes for 21 different amino acids using a triplicate code. That was that slide where I showed you just little 3 base pairs encodes for a particular amino acid. Alleles are different base pairs at different specific portions in the gene. Differences or variants in the alleles account for genotype, and differences in how the gene expressed accounts for phenotype. Differences in alleles or variations in alleles or variations in our genes can be a mutation and cause disease, or it can be a polymorphism that does not cause a non-working protein and therefore does not cause disease. So, like the color of eyes. Okay. Mutations can be inherited, or can occur new, or can occur over our lifetime. Over our lifetime, we develop somatic mutations. All of us have them. Different genetic analyses can detect different types of genetic disease. So, one of the things I didn't cover, and it's very important, is that not even the most sensitive genetic testing, like whole exome sequencing, will not necessarily detect things that can be detected in the microanalysis or the chromosome analysis. So, you certainly don't want us to go, and guys are hopefully you're not being put in the position where you have to order genetic testing and you have the resources to refer your patient, but there is a logical step in ordering specific genetic testing based on the symptoms that someone has. And as I've been saying over and over, genetics will become more and more of everyday healthcare practice. I just want to, there was some stuff last week I didn't cover. I'm going to quickly go over a little bit of that in our time frame, and I'm going to kind of catch up, because at the very end we go over just kind of some of the things for you to think of when you see someone to consider making a genetics referral, and I think that part is important. And you'll get more of that over the summer's lecture, but I just want to go over that. So you guys all remember this from last week of autosomal recessive inheritance, autosomal dominant, and X-linked recessive, and X-linked dominant. And we went over this briefly, but important points of pedigree review: rule out your X-linked if male to male. X-linked transmission does not cause affected males having affected sons. Okay, think X-linked if only males are affected. New mutations can occur in autosomal dominant or autosomal recessive if there's vertical transmission, meaning there's someone affected in every generation, and affected individuals are having affected children. Think autosomal dominant. There are tricks to our inheritance, and this is something I think I went really quickly over last week. There's variable expressivity, so different people can have different symptoms and have the same exact genetic mutation. So here we're looking at neurofibromatosis, and someone has just simple cafe au lait spots and inguinal and axonal freckling, whereas someone else has very severe neurofibromatosis covering her upper trunk of her body. There's age-related penetrance, meaning that you have the genetic mutation, you were born with that genetic mutation, but you don't exhibit symptoms of it until you reach a certain age. Or there's sex-related penetrance. You have both males and females can have that particular mutation, but if you don't have the particular tissue that's susceptible to that mutation, you may not ever display symptoms of it, but you still can have children affected with it. So fathers with BRCA1 and 2 mutations can have daughters with BRCA1 and 2 mutations who go on to develop breast and ovarian cancer. And then there's reduced penetrance. So there are particular autosomal dominant conditions where even if you have the genetic mutation, for reasons we do not yet know, you never develop symptoms of that particular genetic mutation. We're really interested in learning what those reasons are, because hopefully that will help to treat and prevent that in other individuals. I don't think I covered this last week. So anticipation, this is the phenomenon by which the disease severity increases with each generation. And part of this, and for conditions in which they have anticipation, it's caused by having this triplicate repeat expansion. So remember I said when you have a particular set of base pairs that repeats itself over and over, it's particularly susceptible to a genetic abnormality because it's more susceptible to a mistake. If you have repetitive areas, you can have misalignment much easier. So in this particular case, we're looking at myotonic dystrophy, and we're looking at individuals with 2 alleles. So if we look up here, Dad has 2 copies of his particular gene for myotonic dystrophy. He has, and in each one of those alleles, in one copy has 65 repeats of the CTG allele, and he has 5 repeats in the other one. Anything less than 10, or excuse me, anything less than, I believe it's 23 is normal. And so this individual has 2 normal copies. When he has children and passes down that triple group repeat, it expands, and the child is more severely affected with myotonic dystrophy. And by the time he gets down here, we look at a child who has 700 repeats, whereas it started off up here with 65 repeats. And that child is very severely affected in terms of their hypertonia, and their myotonia is much more pronounced. Just kind of a fun fact. So the myotonic dystrophy is referring to the fact myotonia is a process by which you grip and then you try to release and you can't release. So if you ask these people to open a door, they can go and they can open the door, but it takes a moment for their hand to open up to release the doorknob, or to shake your hand, or to tie their shoelaces. So it's a very unique dystrophy. And then in terms of what you are most commonly going to see in terms of disease, these are diseases that have multifactorial inheritance, meaning that there are genes that play an important factor, but there are many other things that play an important factor. And with these particular conditions that have multifactorial inheritance, they're not going to follow that nice Mendelian pedigree. You're going to see that there are interactions between hosted environment, that's simply to mean that my genotype is also influenced by my diet and exercise, my intrauterine exposure, what I do, every day, what am I exposed to in terms of radiation, and other chemicals and so on. And the genetic component is assumed to be polygenic, quantitative, and additive. So there are multiple genes that play an important role. How many particular polymorphisms you have in that particular gene or that family of genes plays a role. And you can have multiple polymorphisms that all kind of push you over the threshold into developing the disease. And things that we think of in this way are many types of birth defects, cleft lip and cleft palates, heart defects. Many of those isolated birth defects are something that push them over within their genetics that put them at increased risk in combination of maybe some intrauterine exposure as well as intrauterine environment as well. And then if you think of in the general population with things like high blood pressure or diabetes, there's a combination of your genetic background as well as your environmental background that are all influencing whether or not you go on to develop that disease. So exactly like I said, environmental, background, major genes. And just to give you an idea of multifactorial inheritance, this is not Mendelian. So if I was born with a heart defect, it's not something like my children have a 50% chance to have a heart defect. It's not one particular gene that I have a mutation in that is causing my heart defect for the vast majority of heart defects. So it's not true for everyone. Some people do have Mendelian factors that cause their heart defect. And one of the ways that this is very obvious is so it's not a single gene. It's also not a completely sporadic event for most individuals. And there is some underlying genetic component because we know this because the risk to have a child with a heart defect is not the general population risk of a heart defect, but it's increased above the general population risk because there's something about my background genetic risk as well as possibly environmental factors. And so for a child with a heart defect, the risk for or a parent with a heart defect, the risk for their child to have one is about 2 to 4%. And there is some relationship that has to do with environmental environments of the maternal environment versus paternal environment. So I'm not even going to go through each one of these, but these are all things that if you think about someone with this condition or heard of this condition, you know that there's a higher likelihood of family history of that condition. So making a genetic referral. So with cancer, really think of cancers at a young age, families, clusters of cancer. That's a reason to make a genetic referral. Cardiac issues, so at this point, it's really things, again, something happening at a younger age or occurring more frequently with a family should be a red flag for you in thinking about making a genetic referral. Any type of neuromuscular disease, any type of cognitive disease, any specific ophthalmological diseases like retinitis pigmentosa, macular degeneration, lens dislocation, that should be hematological diseases there. And so thinking about sickle cell, so you have too much clotting, not enough clotting, those are all things that should make you think about it. Dermatological findings, so calf villi spots when you a referral to genetics. Other things with pregnancy planning, infertility and recurrent miscarriages, a child with an abnormal newborn screen, birth defects, mental retardation, and going on and on. Kids who are having trouble growing, are not meeting their milestones, that have their heads are too big, their heads are too small, unexplained fractures, those all we need a referral to a genetics. And then there's a reason for every pregnant woman to come see a geneticist or a genetic counselor. And it's not because they all need to have something done, but it's because they all need to have their options explained to them. And in this day and age, the amount of testing that you could do during a pregnancy, unfortunately, a OB or a general practitioner is just not given the time frame that they need to have a full discussion with that. You guys have to see like crazy numbers of patients in a day. We have to see maximum 6-7 patients in a day. We have a much longer time to spend with these patients and have time to explain all of their options. I think I'm going to stop there then, because I just shoved a whole bunch of information all at once at you guys. But feel free to give me a call, send me an email if you have particular questions about this subject, or if you have questions and you want to refer someone. I'm very accessible by email. And thank you guys for your time. Good luck this summer.
So I'm just kind of curious, like when you were talking about stem cell, do you guys do research like on that as well, or
There
just
is,
like
I know there's a pediatric, someone in pediatric heme is looking into that, so it is something that is discussed with people, and they are looking into, because I know people with, there are people who do stem cell transplant for
adults?
I don't know. I know it depends on the severity of the disease because there's so many complications that come with stem cell transplant that they need your severity of your disease and your episodes to be much more severe than all the consequences that come with stem cell transplant because there's a high morbidity with stem cell transplant. Yeah. But I think they'll do it in adults.
So like if I was like curious about that and wanted to because I already know I don't need to test, I already know I have to do. Like what are there steps that I can take, or would that be something like I'll just have to find a trial for something like that?
If you write down your email, I will ask around. I know there's someone particularly in the pediatric division that's interested in that. I don't know about the adult capacity, but I'm sure there is. And I'm sure it'll be someone in the heme division.
Yeah,
do you have a hematologist that you're followed by, or?
Yeah, he does not think I should do it, but I'm interested just because
it's worth talking to someone to hear what so you can
right.
understand what are the pros and cons of doing
right.
it.
And he was like, well, if you're interested, at least get babies out of the way just in case, because like I said, I'm just really curious to see what's out there, what's available, what's involved, and then maybe able to make my own decision because I don't feel like.
If you write down your email address, I'll send you an email with what I find out. Yeah. Great. I'll send you an email, even if I don't have any leads, just so you have it.
All right. Yeah. Thank you.
Yeah.
I'll check in on Friday if I feel better than, I still think the senior little baby was, got it. But also, like,
Have a good week, ladies.
Thank you. Bye.
Bye. Hi.
Hello.
You gonna shut it down?
Yes, please. Thank you.
That's where I was doing it. Thank you.